So good morning, good afternoon, and good evening, everyone, depending on where you are joining to us. And a warm welcome to FEBI International Seminar Series. We are very excited to see many familiar faces, but at the same time, many new faces as well. Therefore, if you don't know me, I'm Neriman Yilmaz. I'm a postdoctoral fellow at FEBI, and together with Dr. Marcus Birken, we organize this FEBI International Seminar Series. And today, just before we dive into the seminar, I want to remind you that we want this talk to be interactive. Therefore, please ask your questions during the question session. You can also type them into chat box. And now, without further ado, let's get started. And I'm handing over to Bernard Slippers to introduce our speaker today.
Yeah, I'm in an unfamiliar place. Usually I'm there in the boardroom with everybody and would have liked to do so again today. But my daughter is having a matric dance tonight, so I am here at the venue. So if it looks like I'm in a prison, I'm not quite in a prison. But, you might hear some noises in the background, quite festive here. But I didn't want to miss this opportunity to introduce André. I've, of course, invited André. I've had the fantastic privilege to visit André in April, and in June and July spent 2 fabulous months in André's lab, and therefore learned a lot about his work, enjoyed discussing many of these topics with him, and look forward to listening to him this afternoon and having him speak to a community that is my home community. So maybe just to introduce everybody to André. André is a geneticist. He's at the Technische Universität Braunschweig, where I was, as I've just said. And he also, actually, in his training, has had a plant pathology background. So many of you might know him as a person working on the model system Neurospora crassa. But in his studies in biology at the Freie University of Berlin and the University of Münster in Germany, he received a diploma in biology and a doctorate in botany from the University of Münster. He worked on claviceps and he worked on Gibberella, worked on Fusarium. So understands plant pathogens very well. So in that work that he did, he investigated the molecular basis of pathogenic growth and development of filamentous ascomycetes fungi. But after obtaining his doctorate, he just explained to us why he became, for one, allergic to the one plant he was working on, but also wanted to work on a system where he could test these hypotheses, where many of the methods are established. He moved to the University of California, Berkeley, where he worked with Louise Glass. He was a postdoctoral fellow there and started to investigate the molecular basis of cell-cell fusion using the model fungus Neurospora crassa. Since joining the Technische Universität of Braunschweig, André's laboratory has been focusing on cell-cell communication, so building on that work that he did in California, and the fusion of filamentous fungi in different model systems and plant pathogenic fungi. The group has a long-standing expertise in fungal cell biology, combining these methods with classical, molecular, and chemical genetics. André has recently become interested in fungal interspecies interactions within natural plant-associated microbiomes and the functional role of these microbial communities for plant health. And if I say that, you will understand why I've also enjoyed working with André and his group so much, because those are also topics that interest me and interest many of us in FEBI very well. André has many national and international collaborations, and we're very happy that FEBI is increasingly in that network, and we look forward to building that network more strongly with André and his group and others in the region. And today is part of that process. And André, we really look forward to hearing you talk about the systems that you understand and know so well, and the meaning of that to plant health as well. So all the best. We look forward to listening to you, André.
Perfect. So yeah, Bernard, let me start also. And Neriman and Marcus, thank you so much for the invitation. Even if I'm not there at FEBI yet, I'm really excited about sharing some of our data, some of our story with you guys. And as Bernard said, we are geneticists and we are cell biologists. And so I think that's kind of a different crowd I'm talking today. And so if at some point you get lost or whatever, just, I don't know, leave a little note in the chat and leave your questions, and then we fix this. So yeah, I'm going to communicate to you about communication. And it's as Bernard said, communication in filamentous fungi, intra and interspecies communication. And as Bernard said, I got into this whole business when I started as a postdoc, when I joined the lab of Louis Glass, and we are using a Neurospora crassa, the red bread mold, as a model for studying cell-cell fusion. And now you might wonder, why cell-cell fusion? Oh wait, I'm a little confused about my slides here. Before we get into the whole introduction thing, this is actually my little outline. So what I want to do today is I want to talk about two things. So we have the talk will have two parts. So first I will talk about our Neurospora crassa story. That is basically what we are mostly doing in the lab. And the second story is something novel, which also probably relates more to the things you guys are interested in. And I'm really looking forward to discussing these issues with you. So let's start. I started already that I said, okay, we got into this business of cell-cell fusion. And we are using Neurospora crassa as a model organism. And you might ask now, why would we start cell fusion? And actually, it's kind of surprising. I mean, cell fusion is really basic. It's a really basic mechanism and is involved in the development of basically every higher organism. So cell fusion is really basic. I mean, it starts, if you were, let's say, produced in the natural way. I mean, we started with a cell-cell fusion event between egg and sperm during fertilization. Then later in development, when we build muscles, so if you go to the gym and you're really pumping and your muscles are growing, that is based on cell-cell fusion. Also, there are certain cells in our body which regulate our bones, our skeleton. That's also, and these cells are also formed by cell-cell fusion. It is also involved in the formation of certain organs, such as the placenta, shown here. So the take-home message from this slide is basically cell-cell fusion is a really basic mechanism. But when I got into this business, almost 20 years ago, at that time, it was really nothing known about the genes and proteins mediating this process. So at that time, that was kind of surprising to me, because I thought that we understand all the basic biological mechanisms, but no, we are far from that. And especially in cell fusion, there was nothing known. And at that time, 20 years ago, there was kind of like an international research forming, all using different model organisms to study this event, cell-cell fusion, and to identify genes and proteins which are involved in this process. And as I said, we are using the red mold. And so in the next question you might ask is, okay, when does a fungus fuse? I mean, you probably knew about the things I've shown on the slide here, but I'm not sure if you're all aware that fungi also do fuse. And I'm going to show you a little movie here, and it's showing what we call germ-link fusion in Neurospora crassa. So what's going on there is that we have spores of Neurospora. You know that most filamentous fungi produce vegetative spores, so-called conidia. And when you place these conidia on a petri dish on agar, what they do is they germinate, and they're shown here. So the round structures here are always spores. And they started to make little germination tubes. And when I start this movie, you can see that these tubes recognize each other, they grow towards each other, and they fuse. Shown here, so we have this little group here, a little group here, now another fusion event here. And now these two groups will also be combined. See, they then continue to grow as one unit. So here we have another fusion event. So what's happening there is that these individual spores, which are genetically identical, basically give up their individuality and they merge into some type of functional unit, which then further develops into the mycelial colony. And that's, as you can imagine, a great model to study cell-cell fusion because it is so simple. So the only thing we have to do is we have to harvest conidia, we put them on a petri dish, we wait for 3 to 4 hours, and then we have cell fusion events all over the plate, which we can very easily analyze by microscopy, for example, by fluorescence microscopy, things like this. So it's much, much easier Then a lot of the other model systems, for example, Drosophila, where people study muscle development, where you really have to dissect these little muscles and find the individual cells. So this is very easy here. And actually for the second part of the talk, keep in mind the next thing, which is basically that these cell-cell fusion events are kind of common in a lot of different species. So a friend of mine, Gabriela Roca, did a little literature survey also 15 years ago, and you see that in a lot of different fungi, these germ-link fusion events have been described. Maybe if you look into this list, some of your favorite fungi, I mean, we have botrytis there, we have fusarium there, we have penicillium there, so it's, in general, it's very common. And even in the early days of mycology, it caught already the interest of the early researchers. And you see these beautiful drawings here from the 19th century, where people shown already these fusion events between conidia. So take-home message here: spores of these fungi very often fuse with each other, and so they are really great model organism to study the molecular basis of cell-cell fusion. So what we are interested in, or what we were interested in, and what we are still interested in, is to identify genes and proteins which mediate this process. And as Bernard mentioned at the beginning, we are geneticists. So what we usually start with, so we look for genes, we look for fungal strains where an individual gene is inactivated, is knocked out, so that it has a certain phenotype, and we are interested in the phenotype non-fusion. So that means that these mutants are not able to fuse anymore. So here I'm going to show you 3 of these mutants. So here on the left side, we have our wild type. This is how Neurospora crassa looks. And if you zoom in with the microscope, you see when the colony is established, what I've just shown you in the movie, that these spores make these little bridges here and fuse with each other. And if we look at these 3 mutants here, you see that the spores are still germinating, but they kind of ignore the other spore germ links next to them. They don't interact, they don't form this network, and yeah, sometimes they're almost blind to each other. They don't, it looks like that they don't realize that there are spores around which they could fuse with. And when you can quantify the whole thing by easily just counting all the spores on your plate, and counting how many spores are involved in these fusion events and how are not. And you can very clearly see these mutants have like a 100% phenotype. There is 0 fusion. And MEG1, what are they? Now it's getting super complicated, or it looks a little complicated, and that's now, it gets very molecular, but I try to keep it simple. So these are actually 3 of the first mutants we started working with. So at the time, we only had these 3 mutants. But now we know that many more proteins are involved in this process, and we know that there's actually, so now we know that there's an entire signaling network which is mediating this process. It contains 2 MAP kinase pathways shown in blue here. So MEG2 is a MAP kinase, MEG1 is also a MAP kinase. And as you probably know, MAP kinase is involved in translating signals from outside of the cell to the inside of the cell, also to the nucleus. Then we have 2 signaling complexes here which produce reactive oxygen species, so-called NADPH oxidase complexes. And I'm not going to talk about them today, so you don't have to remember these things. And also a conserved signaling complex, but we don't necessarily know what exactly the proteins are doing. And so another thing we do very routinely is that we use cell biological approaches, and that means that we localize the proteins in the cell. So how do we do this? It's a very common method used in cell biology. We use fluorescent proteins like GFP, the green fluorescent protein, up the protein in question, like in this case here, MEG2 with the green fluorescent protein, and then we do fluorescence microscopy. So that's shown here. So you can see 2 germ links. So here again was a spore, there was a spore. They started to germinate. And these spores express now MEG2, this MAP kinase I was talking about, signaling factor, hooked up to GFP. And as you can see, the entire cell is green. You know, we have a green. We would expect for this type of MAP kinase, because these kinases are always sitting in the cytoplasm, in the nucleus, basically everywhere doing their job. But what you probably also noticed already is that these tips are a little brighter. And as you probably know, I mean, fungi grow at the tip. So when these two spore germlings communicate and try to touch with each other, they grow at the tip, and these tips kind of need to be steered in the right direction. And we see that the signaling factor is sitting at the plasma membrane of these tips. So that was already really promising, that the protein somehow plays a role in steering the cells towards a proper position. But the really great thing about GFP is that you can observe it in living cells. So to image GFP, you do not have to fix the cells, you do not have to kill the cells, you can really look at living cells. So what we call live cell imaging. And when we did live cell imaging of the cell pair here, we found something really interesting. And now please look at the cell tips when I start this little movie. Now you can see that the signal is kind of dynamic. You know, when like the upper tip is lighting up now, the lower tip is dark. When the upper tip is going dark, the lower tip is lighting up. Now the upper tip is lighting up again. And it's going dark again. And it's lighting up again. And it's going dark again. You know, so that was really weird when we saw this for the first time. So we started to looking at a lot of these cell pairs, we quantified it, and we measured also the signal intensity at the cell tips. And we found that basically the MAP kinase oscillates towards the cell tip, towards the membrane. And this oscillation occurs in exact anti-phase in these two cells. So when the signal is up in one cell, it's down in the other one. 6 minutes later, it's up in the second cell and down in the first one. And so on. It continues like this until the two cells make contact and touch. So that was really weird and really interesting. So we had this other mutant. So we were like, okay, how about the other proteins? And as you probably remember, there was also the second mutant in my list of 3 mutants. I didn't talk about this yet, the soft mutant. It's a fungal-specific protein at that time of unknown function. And we also hooked it up to a fluorescent protein, and this time a red one, DS red. And if you just take a look into the central panel here, you can see the same thing is happening. So that's a time course. And you can see at the first time point, we have the signal accumulation at the right tip. A few minutes later, actually, I lost my time labels here. But so this is kind of like 6 to 12 minutes later. You see the signal has disappeared from the first tip and is accumulating at the second tip. Again, 6 to 12 minutes later, it's occurring on the right tip here. So there's also this oscillation and the switch between the two cells. And when we co-localized the whole thing with MEC2, with our second factor, we were in for another surprise because we've seen that both proteins oscillate exactly in antiphase. So when MEC2 is present at one tip, soft is present at the other tip. Next time point is has changed, and soft is present at the first tip, and MEC2 is present at the second tip. So that was a little weird. So what was our interpretation of these data? And our interpretation, actually, it's kind of simple, and it's holding to the last 10 years, basically. All our new data we're finding are supporting this hypothesis. And the hypothesis is that the two cells switch between signal sending and receiving. So what does this mean? We believe that they alternate, that for the first 12 minutes, first cell is sending a signal and the second one is responding to it. And for the next 12 minutes, the second cell is responding and sending a signal and the first one is responding to it. And that's why I also like to call this a cell dialogue, or it's kind of a cellular dialogue. It's really like, like in a good dialogue, you know, where people alternate in talking to each other and alternating in sending signals and receiving signals. You know, what we are doing right now is not really a dialogue. You know, I'm like sitting here in the office in front of my screen, sending out signals. I'm not really sure if you're receiving them or not. Maybe you switched off your computer already because you're like, well, this is all weird. I don't want to hear it anymore. So that's not a good dialogue. You know, a good dialogue would be like two people sitting at the table and alternating in signal sending and receiving. And we believe that this is what these two cells are doing. And that was actually the first time that something like this has been shown for a biological system. So it was like really surprising, really new. And to be honest, I think that was also the reason why I got my job at the University of Braunschweig. They were not looking for a fungal biologist. They were just looking for a geneticist. And I think at the beginning, they were even critical of inviting a fungal person to the job talks. But when they saw the story, they were like, okay, this is unique. This is kind of fun. And of course, we were also really excited about it. And this cell dialogue mechanism is actually the thing we are studying, or we keep studying in my lab for the now last 15 years. But it's we learned a lot of new stuff, and I will give you a few examples. Don't worry, I'm not going too much into molecular details or anything, but I want to give you a little idea about how we're working and also introduce some of the methods. But at this point, your take-home message, everything you have to take from this up to this point is that these germinating spores undergo fusion. They communicate with each other. They fuse, and we believe that their communication is taking place by the cell dialogue, where the cells take turns in signal sending and receiving. So that was the first two mutants I was talking about. If you remember, there was also a third one. Here's a complex picture again, and what I've shown you so far is MEC2, this MAP kinase here, and SOFT, the other factor. But there was also this third factor, this other MAP kinase, MEC1. And probably you remember that the phenotype of all the three mutants looked exactly the same. The cells were not like communicating at all with each other, they were just growing there. So we thought that maybe MEC1 is doing the same thing, you know, also showing this little oscillation thingy. However, it's not. When we localized the protein, we found it did not accumulate at the membrane, at the tips when the cells were growing towards each other. But we found something else which was interesting. We found that MEC1 is only accumulating at these tips here when the two cells touch. So here we have two spore germlings. Again, they are green because we have MEC2 hooked up with, MEC1 hooked up with GFP. And you see they made already contact. Here's still a little space in between if you look very carefully. And at the moment the cells touch, the protein is very strongly accumulating at this point. But here at our next time point, when the cells are fused, it's gone again. So that's also something that indicates that this signaling factor probably plays a role in contact recognition, and not only in the communication before these two cells basically are in physical contact. And again, that's something we would have not thought just by judging from the phenotype. But so now we wanted to study this in more detail. And actually, I'm just showing you this example because I want to introduce you to a really cool new method, which I will show on my next slide. So you can imagine if we use a knockout mutant, we can't analyze the function here after cell-cell contact, because cells which carry a gene knockout of MEC1, which doesn't make this protein anymore, they don't even get to this point. You know, they don't even talk to each other. They don't even get to this point that they touch. So the knockout mutant didn't really help. So we were looking for a way to inactivate the activity of the signaling factor at any given time during the process. So the idea was that we can start the process, let our cells grow, and then we inactivate our MAP kinase. You know, it took us a while to come up with something, but we found something really cool in the yeast literature, and that's shown here. And that's an approach which we also call chemical genetics. I'm not sure if you have ever heard of chemical genetics. When I started these experiments as a postdoc, I didn't have heard about it, but it's a really, really powerful tool, which we are still using a lot in our lab. And that's why I would like to introduce it really quickly. And I'm seeing I'm running already late with my talk, so we will skip some of the coming slides. But I want to introduce this part here because, again, I think it's a really cool method, which is really powerful. So what's the whole basis? So I told you that MEC1 is a MAP kinase, and as you probably know, kinases phosphorylate target proteins. And so to do so, every kinase binds ATP. ATP is a substrate they use where they take the phosphor group to transfer to another protein. And that's shown here. So the green circle is our MAP kinase or our kinase, and it's binding ATP. And so what you can do is you can take an ATP analog, which looks kind of similar to ATP, but is not ATP. And if you give this to any kinase, you will inhibit it, because of this ATP analog will bind into the ATP binding pocket, just kind of shown here, and then inhibit your protein because it's not ATP. So the enzyme cannot really use it. The problem is, however, if you take a general ATP analog and you throw it on your cell, everything is dead, because every single kinase in your cell, every ATP binding protein will be inhibited by this compound. It's dead, so it's super non-specific. And so now the yeast people came up with a really cool idea. So what they did, they took the kinase they wanted to study and introduced a point mutation. And by introducing this point mutation, they enlarged the ATP binding pocket. So they made it bigger. And that's indicated here that we have like this white space here. So there's empty space. And this enlarged ATP binding pocket can still bind ATP. So it's still functional in general. However, now if you add another ATP analog, which is really big, and this ATP analog is so big that it doesn't fit into any natural occurring ATP binding pocket. So if you throw it on a wild type cell, none of your kinases will be inhibited because this inhibitor is too big to enter this ATP binding pocket. However, this enlarged ATP binding pocket in your mutated kinase is big enough to basically bind this bulky inhibitor. And by this, you can very specifically inhibit this kinase. And this inhibition takes place within basically 5 minutes. You know, you add the chemical to your cells, and within 5 minutes, you can see that something is happening. And again, it's very specific because this inhibitor is so big that it doesn't inhibit any of your other kinases. So we did this for MEC1. So we constructed a strain. And I mean, that's why it's called chemical genetics. We combine genetics, so the genetic introduction of this point mutation, with a chemical inhibitor. So we constructed the strain, and that's shown here in a little quantification on the right side. So here, the first one with 0, we have our strain, which is expressing this kinase, and we add 0 of our inhibitor, and 100% of our germ-link pairs look normal. So blue means normal, they grow towards each other, they touch, and it's all normal. If we add a very high amount of our inhibitor, like 500 micromoles here, the phenotype looks exactly as in the gene knockout mutant. You know, the kinase is fully inhibited and the cells do not even interact with each other anymore. However, if we add kind of an intermediate concentration, like 100 micromole, we see that the cells still interact, and 1/3 of them still look normal, but 2/3 of them develop a new phenotype. And that's what we call the twisted phenotype. Now you might ask, what's that? Well, I posted a picture here. And here you can see 2 spore germ-links. So here we had the spore body and they germinated. You see they made contact, but now they build kind of a weird structure which looks like a corkscrew. You know, it looks like as if they grow around each other. And we were really curious to see what's happening there. So we did again do live cell imaging. So here we have like 2 cells and they grow on this inhibitor. So the inhibitor has already been added. And when I start this movie, you see they still grow towards each other, they make contact, and now they would usually fuse. However, these guys do not fuse. As you see, they continue to grow. But the weird thing is they also stay in touch. So it looks as if they're still attracting each other, but they don't stop and they don't fuse, but they continue to grow. And since they're still attracting each other, they kind of turn around each other. You know, it's almost like a romantic comedy. You know, you have like these 2 partners, they have met already, everything is fine. And then they start all these unnecessary complications and they, you know, turn around each other and do these weird things and do not fuse. And And this phenotype now fits really well to what we observed in the localization of this protein. If you remember, I've shown you that this MAP kinase only accumulates at the cell tips once the two cells touch. And that was kind of an indication that this MAP kinase also plays a role in cell recognition, so that the cells notice and realize that they reach their partner cells and that they have to switch their program now from directed growth towards fusion. I kind of included this part in the talk because I have to say it's one of my favorite phenotypes. And again, you know, I want to give you guys an idea how we're working, what methods we are using, and I thought so maybe it's a better idea to show you some of these bits and pieces and not go into all the molecular signaling networks we are known, which would probably a little bit too much. So now in the next part, I wanted to present some new data on signal emission. So we learned a little bit more about how the cells are communicating with each other and how they emit their signal. But let me check the clock. I'm already talking for like 35 minutes. So probably we are going to skip that part. So what do I want you to take from this first part of the talk? So the things you really should remember is, as I said, Neurospora crassa employs like this weird dialogue-like signaling mechanism in germline fusion or in vegetative cell-cell fusion. And the signaling is mediated by a conserved signaling network, which includes these two MAP kinase modules. And you've seen that these MAP kinase modules undergo really high dynamics within the cell during the process, so that they oscillate towards the membrane, are released from the membrane again, and it's all highly dynamic. The last 2 points here were like the stuff I skipped now, and it's really no problem that we skipped it. It's kind of like, I had it in the talk kind of like as a little joker thing, you know, if I would have been very quick with everything I told you, we would have time for this, but it's no problem that we now switch to the second part of this talk. And again, I hope that so far that you could follow, that it wasn't like too molecular and that you got lost there. I think that's always a challenge with these online talks. You know, if you would be sitting together in one room, I could see your faces, you know, and I would see if you're like nodding and you're like, okay, we understand what he's talking about, or if your faces would be like really like, what? What is he talking about? So I hope you could follow so far, and now we can dive into our new topic, which is actually interspecies communication. So communication between two different species, because everything I've shown you before was like communication and cell fusion within one species, Neurospora crassa. And that second topic here actually also started with a question. And it started with a question which I always got after presenting the data I've just shown you. You know, every conference, every meeting I went where I would show this, people would always ask, or people would always say, they're like, you know what, it looks really cool, it's really interesting, but is it conserved in other fungi? Do all fungi do this? Or is it something which is like, yeah, let's say super specific for Neurospora? And to be honest, I mean, that's an important question. Because I mean, if it would be only specific for Neurospora, it would be still an interesting biological phenomenon. But and maybe you could say it's not so relevant if it's only happening in this model fungus and in no other fungi. And so I was always hoping that that other people would take on this topic and check it in their fungi. And some people tried, but then they struggled a little bit with the cell biology. And I mean, the cell biology always looks super easy, you know, when we show these movies and when we show GFP, it all looks like very simple. But honestly, it's not. I mean, establishing these methods for a new fungus and working out the microscopy and everything is sometimes a little challenging. And so it didn't really happen. And so in the end, I got a little tired and I was like, come on, then we just do it by ourselves. So we had to choose a different fungus. And I wanted a fungus which is not too closely related to Neurospora. you know, because we wanted to see if it's like broadly conserved in fungi. And I also wanted a pathogen. Why and why is that? The proteins I just mentioned to you, you know, these two MAP kinases, or even all these factors I showed you in this little cartoon, you know, when I talked about like the signaling network, all these different factors there. It has been shown for a lot of fungi that all of these factors are also involved in pathogenicity, you know, also in plant pathogenicity. So it seemed as if there's some relationship, some molecular relationship between the cell-cell fusion events and yeah, let's say pathogenic growth, pathogenic development. And so that's why I also wanted to have a pathogen so that maybe we can relate these things to each other. And so we chose Botrytis cinerea, and probably all of you or most of you know Botrytis cinerea. It's a very broad pathogen. It has a very broad host range. So in Germany, it's a big deal on strawberries. That's why I brought this little photo of strawberry here. But it's also a big issue on grape, on all types of vegetables, on flowers. So it's a really important pathogen. And so the first thing we did is we checked for germ-link fusion. Does the fungus do it? And yes, it does. So here you can see spores of Botrytis germinating, and you see they also make contact. They also fuse with each other, very similar to Neurospora. Then we made mutants for the genes I've introduced to you, like SOFT and MEC2. So SOFT is called, in Botrytis is called Pro40, and the MAP kinase is called BMP1. And so here on the right side, you see a little quantification of these fusion events. So the dark bars are also always germination to show that the spores are healthy, that they germinate. And in the wild type, if you look at a spot at a population of spores, you see that almost 50% of the spores undergo fusion. When we knock out the soft homologous gene, we see it's down to 0. When we knock out the BMP1, the MAP kinase encoding gene, it's also down to 0. And these are just controls where we reintroduce the wild type gene and we see that it's complementing the mutant. So this phenotype is really caused by this single gene knockout. So at this point, we can say, okay, looks also very similar. So spores, germinating spores undergo fusion, and these two proteins are also involved in it. We also need it. However, to really prove that the cell dialogue thing is going on, we needed to do, again, cell biology. So we did the same thing we did in Neurospora, and we hooked up the two proteins with GFP. And we localized it, we did live cell imaging, and indeed we could find the same thing. Sorry, struggling with my magic mouse again. So we could see the same thing here in the upper panel. You can see the MAP kinase fused with GFP. And you see at time point 0, the protein is localizing at this upper tip. At time point 6, it disappeared from that tip and is accumulating at the lower tip. Time point 12, we have the upper tip again, and then it's accumulating at the fusion point. For the soft homologous protein, same thing, localizing to the right tip, localizing to the left tip, and accumulating at the fusion point. And when we co-localize both proteins, we also found the same thing. So based on these data, we said, okay, I believe we can really say that the cell dialogue mechanism is conserved in Botrytis cinerea. And what I haven't mentioned yet is how close Botrytis and Neurospora are related. You know, at the beginning, I told you we chose a fungus which is not too closely related to Neurospora, but how far are they really? And they're actually kind of far away from each other. So if you look in terms of evolution and times of evolution, it's similar to human and fish. So similar to men and salmon, basically. And I think in light of this, it's even more surprising that this mechanism is so well conserved and that that both species basically show this. And in this paper, which we published last year, we also had a collaboration with Reinhard Fischer's group from the University of Karlsruhe in Germany. And they looked at the same thing in Atrobotris, a nematode-trapping fungus, and found the same thing. So in Neurospora, in Botrytis, in Atrobotris, so the cell dialogue seems to be really conserved or highly conserved in filamentous ascomycete fungi. So that's another take-home message for you to take from this. And now since what I said, since all of this mechanism is so highly conserved, one of my graduate students had kind of like this crazy idea. He was like, what happens if we throw these two fungi together? And what was the motivation behind this? I mean, I haven't told you yet that we don't know yet what the signal is. I'm always talking about, oh, the cells obviously send out a signal, they talk to each other, and I mean, the signal is probably a chemical they are like secreting, but we don't know what the signal is. And so we wondered, is the mechanism in itself is conserved, is the signal also conserved? And since we don't know what it is, we had no idea what to look for. And so the graduate student had this really cool idea. That was Martin Weichert. And he had the idea, he just said, let's just throw the two fungi together. If they use the same signal, we should be able to see that they talk to each other. If it's a different signal, they're not going to talk to each other. And my expectation was like, of course, they're not talking to each other. It's like man and fish. They should have their own signals. However, when we did the experiment, we were in for another big surprise, and we saw that the two different fungi really talk to each other and attract each other. So here, if you look at these images in the left upper corner here, the colored cell in green and red is always Neurospora, and the non-labeled cell is Botrytis. And I think here you can very clearly see that these two cells react to each other and make contact, and Botrytis is making a little side branch here growing towards Neurospora. They also make contact, they touch here. And this is actually very common. When we look in this mixed population of Neurospora and Botrytis cells, we find the same number of Neurospora, Botrytis, and Botrytis Neurospora interactions. So there's not even really a bias towards like intra-species. However, and that's important now, when these two cells touch, they still recognize that they made contact, not like what I've shown you before when we inhibited the MAP kinases where they made these little corkscrews where they didn't recognize cell-cell contact. So the cell-cell contact signal also seems to be conserved, but we never see fusion between these two cells. And I mean, that really makes sense because, I mean, if we would see a lot of interspecies fusion, that I don't even know what the consequences would be. That would be probably all end up in a big mess. So once the two cells are in contact, they realize, oh, it's the wrong one, and they are not going that they're not going to fuse. So take-home message from this slide is there seems to be a universal signal used by these different fungi. Or you could also say, if you want to stay with this metaphor, that fungi probably have kind of like a universal chemical language and that they are able to communicate and talk. And I will come back to this in a second. But before we talk more about the interspecies thing, wait, this we can skip. Oh, wait, yeah, that would have been probably your next question. I almost forgot about it. Of course, there's also the question, is also the cell dialogue mechanism mediating this interaction? And indeed it does. So we localize the proteins again. So we have the MAP kinase, and the green one is Botrytis, and the non-colored one is Neurospora. And you see that the MAP kinase is also oscillating in this case. So that means that also these two cells, which are as distantly related as man and fish, are able to coordinate their behavior over the spatial distance and get into this dialogue kind of communication. And yeah, to be honest, I think that's pretty amazing. Two different species have not even made contact. It's basically all based on secreted signals, something like this, and they are able to coordinate their behavior. And here we did a colocalization again. I'm not going into all the details, just trust me, it's also the dialogue mechanism which is mediating this interspecies interaction. But now, before I continue with this, we found another interesting thing in Botrytis, and in the end I will all get it together. So what was the other interesting thing in Botrytis when we talk about germline fusion? And since it's a plant pathogen, so we had this is like an old paper from like more than 10 years ago, where we published that botrytis is also doing this fusion thing. And since it's a plant pathogenic fungus, I was like, well, we should also visualize fusion on the plant surface to show that it's also taking place during the normal growth conditions of botrytis. And since it's a little bit difficult to do microscopy directly on for example, on the leaf, on a strawberry leaf or something, because all the plant tissue and it's all the chlorophyll in it and everything, it's a little challenging. We used a little trick, which is kind of well established in cell biology when you work with plant pathogenic fungi. It's a little trick where you imitate a plant surface by using an onion epidermal layer. So what is an onion epidermal layer? Probably I should have brought a slide. I hope you get it even by listening to my voice from the off now. But it's mine. If you're in your kitchen and if you peel an onion, you probably all know this thing that you can peel off this very thin layer, this very kind of little skin of the onion, which is basically just one layer thick. And I don't know, at least in Germany, that's like what kids do in school. It's the first thing they do microscopy on. It's like this onion epidermal layer, you pull it off, put it in water, put a little ink on it, and then you can see nicely the plant cells sitting there. And so what you can also do is you can peel off this onion epidermal layer and put it on a petri dish on an agar plate, and then put the fungus on it. And so the fungus recognizes that it's sitting on a plant surface. And usually, or a lot of plant pathogenic fungi then start their infectious growth, make aprosoria or types of things, and penetrate this plant layer. And so we started these experiments here. And when we did these experiments, we noticed something which I didn't know before, I have to admit, that this onion epidermal layer has two different sides. One side is hydrophobic and one side is hydrophilic. And to our surprise, we found that there's a difference if we place the botrytis cells on the hydrophobic or on the hydrophilic side of this onion epidermal layer. So here, when we put it on the hydrophilic side, we got cell-cell fusion. That is our gray bar here. Very similar to what we see when we do it on just minimal medium on our agar. That was all the stuff I've shown you before, was on minimal medium on agar. So hydrophilic side of the onion epidermal layer, exactly the same thing. However, on the hydrophobic side, no fusion at all. But we found that on the hydrophobic side, and that's shown here, the fungus is penetrating the onion epidermal layer and is basically infectious. And so from that, we basically came up with the hypothesis that botrytis is kind of making a developmental decision depending on the signals it's receiving from its environment. So if it's sitting on a hydrophobic surface, which usually indicates I'm sitting on a plant, because plant surfaces, like plant leaves or on needles or whatever, are usually hydrophobic, then the fungus is penetrating and it's infectious. But then at that time, it's not fusing. So it was kind of the idea that fusion and infection are basically kind of mutually exclusive. So then we thought, well, wouldn't it be cool if we could have them on the hydrophobic layer and add the fusion signal and see if we can reprogram the fungus, or we can kind of disturb it. But as you remember, I just told you, we don't know what the signal is. We don't know this chemical. But what we do know is that Neurospora and botrytis are talking to each other. Now I'm getting back to interspecies. So we checked Neurospora and we found that Neurospora doesn't care if it's hydrophobic or hydrophilic. Neurospora is always making this fusion. So then we said, okay, let's take the hydrophobic event and put Neurospora and botrytis together. So the idea was botrytis would usually go into its infection program, but Neurospora would provide fusion signals. And what did we find? We found indeed that if Neurospora is there on the hydrophobic layer, we do see that botrytis is now fusing again. Without Neurospora, zero fusion. That's also shown here. The light bar is fusion, zero fusion. If we add Neurospora to the mix, botrytis is fusing, and botrytis and Neurospora are also interacting. So it seems as if when these two fungi are sitting together on a plant surface, one fungus is reprogramming the developmental decision of the other one. So Neurospora is reprogramming botrytis. And as I said, we believe that infectious growth and fusion is kind of mutually exclusive. So from that, we came up with the hypothesis. We said, well, if Neurospora is there, and if Neurospora is reprogramming botrytis, maybe botrytis is less pathogenic. And we did a pathogenicity assay. So here this is not strawberry, this is bean, like just green bean. So we took like leaves of green bean. When you infect them with botrytis alone, you see that after a couple of days, you get like this big necrotic area indicating that the fungus is infecting. When you add Neurospora alone on it, nothing is happening because Neurospora is not pathogenic on bean. And interestingly, when we mix these two fungi, botrytis is also not really pathogenic anymore. And that's also shown here in a quantitative analysis. So that's like the diameter of the necrotic area. So the big bar here is botrytis by itself. And here we mix Neurospora and also different Neurospora mutants into this mix. And you see that the presence of Neurospora is suppressing the pathogenic development of botrytis. And so initially we wondered, well, maybe Neurospora is just killing botrytis or something on the bean leaf. So we did microscopy on it. So here is botrytis alone, and you can nicely see how it's making its little infection structures. And here you can see botrytis and Neurospora together, and botrytis is still happy, it's still growing, it's germinating, it's not dead, but it's making fusion, which we didn't see before. And wait, now I'm going the wrong direction. I should come to my conclusion now, wait. Okay, so there are like some extra slides in there. Yeah, and if I look at my watch, I think it's time to come to a conclusion. So what did we learn from all of this? So that was now the part with interspecies interactions. And the first thing we learned, the cell dialogue mechanism is conserved in filamentous fungi, and I would say highly conserved. It's also mediating interspecies interaction. So the so far unknown signals and receptors are conserved. There seems to be a common molecular language. And really important, and I think that's really, really important for the things we want to do in the future, the presence of one fungus can change the developmental decision of another one. And so why is this important? I mean, what I've shown you so far about botrytis and Neurospora, you might argue and say, okay, that's kind of artificial. You know, you do this in the lab, you mix these two fungi, but probably to be honest, I mean, Neurospora and botrytis probably never meet in nature. They probably don't grow in the same habitat. However, since we saw such a strong influence of fungus on the other, what we now want to study and what we started to study is the behavior of fungi within natural occurring microbiomes, so natural occurring fungal communities. And we believe that within these communities, there's also obviously communication between these different species going on. And probably they also really strongly influence the developmental decisions, like one fungus of the other one. And not much is known about this. And I think it has really a lot of potential to study these things, understand these things. I mean, you might argue maybe in the end we could come up even with a totally new way of biocontrol of pathogenic fungi just by establishing microbiomes which contain fungi which can control and reprogram these dangerous species, and maybe not by killing them, but by reprogramming their developmental decision. So now I really talked a lot. So now I just want to finish my slide. And you know, when I got confused about my other slides, I realized that this is an old slide. And so here's my group, here are the collaborations, and you see what's missing. Fabi is missing and Bernard is missing. And that's really unforgivable at a talk like this. So of course, there needs to be a really big label with Bernard and Fabi, because I mean, we want to venture together into these new questions we are asking, into the questions of microbiomes. And I'm really excited about meeting Bernard at the time when we were thinking about these things, and because he's really an expert on all of these things. And I think Fabi It's an amazing institution to ask these questions, and that's why I'm so grateful that I could give my talk today, that we can discuss this. And yeah, I'm looking forward to all the things we are going to do in the future together. And now time's really up. So thank you so much for your patience. And I hope maybe that there's a little bit time left for questions.
I hope you hear our interaction with the clapping, André. And I'm handing it over again to Bernard for the question and answers. Or Bernard, do you want me to do it?
Yeah, André, thank you very much. It's I've heard the story. We've discussed these things before, but it's as exciting when you present it again here. So thank you very much for the time and presenting it to the community. I see already a number of questions there in the chat. André, your video has just gone off.
Okay.
I don't know if you're still there.
Maybe I should. Oh wait, yeah. Wait one second.
And maybe we start with those questions in the chat box. I'll just read the first one there from Andy Wilson, who's currently in Denmark. She's a Fabian that's still interacting with us a lot. She says, "Thank you for an absolutely phenomenal seminar, André. I really enjoyed it. Have you looked into any of the cell fusion events that happen during sexual reproduction in the ROSPA, and whether you see similar defect with the MAC1, MAC2, and so mutants? Specifically because MAC1 and MAC2 are really important for mating and regulating perithecium development."
Yes, that is a really, really good question. And we started looking into these things, and it's way more difficult in terms of studying it. So Neurospora is making little fruiting bodies, and from these fruiting body, female receptive hyphae, a so-called trichogyne, is emerging, which is also responding to a pheromone gradient, where to look for a basically mating partner. And during mating, it doesn't seem to be like a bidirectional thing, but a unidirectional thing. So the male, which could also be just a spore, is basically sending out pheromones, and then the trichogyne is going there. So it's not a dialogue, it's more like a monologue type of thing. And you're absolutely right. I mean, there is this big question of the role of these MAP kinases in this process, especially since it's amazingly well established in yeast. You know that the MAP kinases are doing the pheromone response, and actually our MAC2 MAP kinase I was introducing today, the one which is doing the oscillation, is the homologue of the pheromone response pathway. The problem with studying this is, again, that these mutants have a very early phenotype. So like these delta MAC2, delta MAC1 knockout mutants, they don't even form these fruiting bodies, and they don't even form these trichogynes. So you can't really check, or you can't check in the mutant if it's involved in like the directional growth of the trichogyne. But what we want to do now is we want to use this chemical genetics approach again. So the idea would be, you know, that we grow them without the inhibitor until they make trichogynes, everything. Then we add the male partner. Then we add our inhibitor, and then we see if the trichogynes are still able to grow there. And to be honest, I'm asking my graduate students for a long time to do these experiments, but for all of them, it's just a side project. And I know from my own work, it sounds simple, but it's technically challenging. So no one has really taken the time to going into all of these details, but I hope we have this answer soon, because as you said, it's a really good question, and I personally also really want to know it. So to make it short, we're working on it. Yeah.
All right, thank you, André. Quentin Santana is asking, "Thank you for a wonderful talk. Does MAC-SO signaling interaction occur on vegetative cells that fuse, or is the signaling specific for germing tubes?"
No, it's basically involved in different types of vegetative fusion. I mean, we have another one which is hyphofusion. So what I've shown you today is like the germlings, and I mean, they do the thing when the colony is established. And once a colony is established, also within these mature colonies, there is cell-cell fusion, because between the long leading hyphae, cross connections are formed by branches which grow towards each other and fuse. And we also studied this, and it's the same mechanism. So they also use this cell dialogue mechanism, the same proteins are involved, and it seems to be the same thing. And so I briefly mentioned our colleague, Reinhard Fischer, who studied Atrobotris, and you guys probably know Atrobotris is making these ring traps to catch nematodes. You know, it's making these little loops, and they are sticky, and nematodes grow in or walk in and get basically caught. And in order to make this loop, hyphae also have to fuse. You know, you have a hyphae, it's making a branch, and it's growing towards the hyphae again, and then they fuse. And even that fusion event is also mediated by the cell dialog mechanism. So our interpretation of this is that it's a very, I almost said like an old mechanism, a really highly conserved mechanism, which is employed in all types of different developmental processes which involve vegetative cell-cell fusion. Yeah, but seems to be really conserved.
Thank you, Andre. There's a couple of expressions of appreciation there in the chat for a great talk. Professor Lucy Molaleki, she has two questions. She asks, the first one, what was the final conclusion on the role of cell-to-cell communication in pathogenicity or virulence of botrytis? And the second question, in bacteria, there are two types of signaling, one that is generic to all bacteria, autoinducer 1, and some more specific for closely related bacteria. Do you think the same occurs in fungi?
Yeah. So the first question is really, what is the conclusion in terms of the role of germline fusion or hyphal fusion in pathogenicity? And I have to say there is no clear I just want to say confusion. That would have been a Freudian slip, yeah, because there's some confusion. It's still not clear. So we know that the same proteins, genes and proteins are involved. You know, when I initially showed you this cartoon with this entire signaling network, we now know that the signaling network is involved in a lot of different developmental processes of fungi. So it's almost kind of like a molecular brain, you know, where like all types of different signals from the environment go through this through the signaling network and to elicit the appropriate response. But there's no clear relationship that you can say if they can fuse, they are pathogenic, and if the mutants which cannot fuse, they are not pathogenic anymore. There are some publications where people kind of give the impression, but I was always highly critical of this because, I mean, it's difficult to distinguish in these mutants. Maybe if you take out this protein, you know, maybe you also take out the specific function this protein has in pathogenicity, and it's not directly related to fusion. And so it's yeah, I'm wobbling around. So it's there is no clear correlation that you can say if they fuse they are more pathogenic or not. That's the one thing. The other thing with bacteria, where we have like general signals and specific signals, it might be similar here. I mean, there seems to be like this general signal mediating this interaction. And actually, it was kind of interesting when we were studying these interspecies things, our conclusion was it's all super non-specific. You know, and we also looked at other fungal pairings, and we see that a lot of fungi do this. But at the same time, my old lab at UC Berkeley, Louise Glass, she was studying different subpopulations of Neurospora crassa. And they found all of these things that some members of these subpopulations could do it with each other, others wouldn't. And their data were like, it looked like it's super specific. And it was kind of like, you know, I remember one meeting in Spain, and we were sitting there and we were like, how do we get this together? My conclusion is it's totally non-specific. Yours is it's totally specific. And in the end, when we all put it together, we realized what our hypothesis is now is that probably it's very difficult in evolution to change the signal and the receptor. However, we still see that there's specificity. And probably what has happened is that basically downstream of these initial signaling and communication thing, they have downstream mechanisms evolved which allow specificity. And this could be, again, specific signals. We know that it's also, there might be something like quorum sensing going on, because we see that these fusion events are also depend on spore density, and that seems to be a different signal than the communication signal. So we believe that there is this really highly conserved thing, but after that, specificity has evolved, or mechanisms has evolved, which mediate specificity. And to be honest, I'm not sure if it's a bit similar to what's happening in bacteria, but it's this whole specificity, non-specificity thing is a really huge deal, and we still don't understand everything of it. And I believe we really need to identify the signal to answer this question in the end.
Thanks, Andre. I think there's one more question I'll ask, but I noted Hamza is with us on the call as well. And earlier nice. Hamza. And earlier he shared the link to the paper, which somebody asked for. So thanks for that, Hamza, and thanks for joining. I'll ask, there's a last question in the chat from Dr. Rajesh Pandai, is from the Botany Bunkulhand University in India. So he was asking, where's that question? Is the cell-to-cell communication an aggravating to pathogenicity in fungi?
Yeah, as I said, we don't really know. And it's really difficult, what I mentioned before, to answer these questions, because once you work with gene knockout mutants, you know, it's really difficult, and you see like that there's kind of like that they are less virulent, and that there's a defect in virulence. It's really difficult to distinguish if this is a cause by the lack of fusion, or if this is a cause by, or that this protein has like the second role, not only in fusion, but also in pathogenicity. It's almost impossible to decipher this. And I mean, one idea, what we believe, people often ask this question, why do they even fuse? What's the benefit? And I mean, and one of the benefits in a mycelial colony is probably that if you make this really nice interconnected network, that it's much easier to move around nutrients throughout your colony. And I could imagine, for example, that in pathogenic fungi, especially ones which like maybe biotrophic ones, which form haustoria, which get their nutrients through haustoria, you know, from like one little source, and they really need to take up these nutrients and send them throughout their entire colony. And there I could imagine that it would be a disadvantage if you cannot fuse, if you cannot really make this proper network. But again, there are, at least not that I know of, there are really clear, convincing data showing this. And mainly because of this problem to distinguish these primary and secondary effects from these gene knockouts. Yeah. But it's a really good question. I would love to be able to answer this question. Yeah.
Great. Andre, thanks a lot. I'm going to bring it to a close there. I see we're starting to lose some people now. I guess people are moving on to the next things. But yeah, you can see there in the profile, I think people are really excited, as I was when you explained these mechanisms to me. And I see people writing messages here to me saying, you know, they're really excited for what you've shared. And I'm really excited about building on this. You know, you very kindly say that there should be a FAG logo there, but so far we haven't contributed enough to be on that last slide yet. But we hope to do so going forward and to explore with you what these cell-to-cell communications and these mechanisms might mean for plant-infecting fungi and in fungal communities. I think that's a really interesting frontier for us to be exploring in the next couple of years. So thank you so much for the time today. For everybody who's enjoyed the talk today, Andre is coming to FABI in November and will be with us so people could meet with him, sit with him, and discuss their work and the work that we look forward to doing going forward. Thank you all for joining us, and especially Neriman and Marcus, who always puts this together for us. And I know Nam has been helping to make sure all the technical things are working today. So a big thank you to the team back at FABI. And I wish you all a great day. And thanks again, Andre. Please send my regards to everybody in the lab.
Well, thank you for having me. And I'm very much looking forward to meeting all of you. This will be really fun. Thank you. And
a big thank you from us as well. Thank you, Bernard, for inviting Andre and basically sending him the right signal to connect with us. Because as FABI, we know that there will be more connection there. And we are very much looking forward. And everyone also joining to our seminars, thank you so much and hope to see you all next time.